Next chapter we are going to discuss is antigen. So what is an antigen? So antigen, it is a substance that when introduced in the body, it triggers the production of specific antibodies. So substances that are capable of inducing immune response. So they can recognized by surface B cells or by the T cells. So they can either be recognized by the B cell receptors or the T cell receptors. So they can bind with the antibodies or the T cell receptors. Now what do you mean by immunogenicity? So the ability to induce a humoral or cell mediated immune response. In summary, antigens, they are the substance that can trigger an immune response and can interact with the antibodies, B cell receptors and the T cell receptors. Immunogens, they are the subset of the antigens that can induce a detectable immune response. Antigenicity, it refers to the ability to interact with the immune receptors, whereas the immunogenicity, it's the ability to provoke an immune response. Now, how do we classify the antigen? We can classify it into complete antigen or hapten. So the complete antigen, they are the substance that can induce antibody formation by themselves and can react specifically with these antibodies. So the complete antigen, they have two key attributes. One is the immunogenicity and other is immunological reaction. So they can induce the formation of antibodies by themselves. When introduced into body, they produce a specific and observable reaction with the antibodies generated. What are haptens? Haptens are substances that they themselves are incapable of inducing antibody formation, so that they lack immunogenicity. Haptens, they become immunogenic, that is capable of inducing antibody formation when they combine with the larger molecule, with the large carrier molecule. So with the large molecule they can bind. So alone they cannot produce an immune response, but with the carrier molecule, large carrier molecules, that results in production of the response. So large carrier molecules, particularly the proteins, that is albumin and globin. Now what are the epitopes? So epitope is known as antigenic determinant. So antigen determinant, it is the epitope. So it is the smallest unit of antigenicity. So the smallest unit of antigenicity is the epitope. So it is a small area on the antigen that consists of 4 or 5 amino acid or monosaccharide residue. So epitopes, they possess specific characteristic including a distinct chemical structure, electrical charge and steric configuration. So it is a part of macromolecule that is recognized by the immune cells. So they have capacity to sensitize an immune cells and react with the complementary site on the specific antibody or T cell receptor. Now what are paratope? Paratope part of the antibody. Paratope is the part of antibody that recognize the epitope. So combining area on the antibody molecule that corresponds to epitope, it is known as paratope. So the paratope on the antibody, it will interact specifically with the epitope on the antigen, contributing to specificity of the immunological reaction. So many antigens such as bacteria or viruses, they carry various type of epitopes. So cross reactivity in the immunology can occur when different antigens share the same or similar epitope. So that can lead to immune response against the antigen that are not the primary target. Now what are the factors that influence whether a substance can function as an antigen in the immune system? They are the number one is the size. The antigenicity, it is related to the molecular size. So larger the molecular size, they are highly antigenic. So particle with the low molecular weight, they are non-antigenic or have weak antigenicity. Second is the chemical nature. Natural occurring antigens, they are often protein and polysaccharides, which tends to be highly antigenic. Lipids and the nucleic acid, they have a lower antigenicity. The protein that is composed of around 20 different amino acids, it tends to be better antigens than the polysaccharides, which have fewer monosaccharides unit. The third is the susceptibility to tissue enzymes. Only substances that are metabolized and susceptible to action of tissue enzymes behave as antigens. So antigens introduced into body, they are broken down the host into fragments with the antigenic determinants. So the phagocytes and the intracellular enzymes will play an important role in this breakdown. So the synthetic polypeptide that are not metabolized in body are not antigenic, while polypeptides, they are antigenic. So the immune response typically does not respond to an individual own normal constituent antigen. So the antigenicity, it is influenced by factors like size, chemical nature, susceptibility to tissue enzymes, and foreignness to the individual. Other are the autoantigens. What are autoantigens? Autoantigens, they are the group of protein or molecules that are naturally present in patients' own body. So under normal circumstances, these self-antigens will not trigger an immune response because the immune system recognizes them as part of body. In case of autoimmune disease, the immune system loses its ability to tolerate these self-antigens and mistakenly mount an immune response against them. So this immune response results in body attacking its own tissues and organs. That causes the characteristic symptoms and the damage that is seen in the autoimmune disease. What are tumor antigens? They are also known as neoantigens. Tumor antigens are specific antigens presented by the cancer cells, and they are not normally found on normal healthy cells. So these antigens, they are referred as tumor specific antigens. The tumor antigens, they are typically displayed on the surface of tumor cells. Other are the heterophile antigens. So these refer to the antigens that are same or closely related, occurring in different biological species, class or kingdom. So the example of heterophile antigens, they are the Forceman antigen. So these heterophile antigens, they can be used in diagnostic serology, like in case with the Weil-Felix reaction that is used in diagnosis of typhus, that involves the use of this heterophile antigen. So the antibodies that are produced during the course of typhus infection, they cross react with these antigen. Other is the Paul Bunel test. So you should know for entrance exam that this test, it is used in diagnostic of infectious mononucleosis. One question that you can get is Paul Bunel test, it is used for infectious mononucleosis. Other question you can get is it relies on the heterophile antigen to detect the presence of antibodies specific to the Epstein-Barr virus. Other test is cold agglutinin test that is used in for the atypical pneumonia. So it uses the heterophile antigens to detect the cold agglutinins, that are the antibodies that can agglutinate the red blood cells at lower temperature. Now what are the super antigens? Super antigens, they are the unique protein molecule like Staphylococcus enterotoxin. The example of super toxin is Staph enterotoxin. So that has ability to activate a large number of T cells. Without considering their usual antigenic specificity. So the key points about the super antigens, they are there is broad activation of T cells, unlike a typical antigen that activates specific T cells with matching receptor. Super antigen would activate a vast number of T cells. Super antigen interact differently with the T cell receptor compared to the conventional antigen. So remember, the super antigen, it will attach to the lateral surface of T cell receptor rather than the antigen binding group. So it acts on the lateral surface. Super antigen produced by microbes, they are typically medium sized proteins with molecular weight that range from 22 to 29 kilodalton. Super antigen triggers the release of certain cytokines like the interleukin 2 from the T cells. So the action of super antigen by causing the release of interleukin 2, it leads to extensive production, proliferation of the T lymphocytes. So that means a large number of T cells, it multiply rapidly. So as a consequence of massive T cell proliferation, a variety of cytokines, they are further released into the immune system. The next is innate immune system recognition. The innate immune system, it recognizes unique molecular proteins that are common amongst various pathogens. The pathogen associated molecular protein, that is PAMP, they are the broad molecules found on the surface of pathogen. They are recognized by the innate immune system as indicator of invading pathogen. Other are the pathogen recognition receptors. So these are the receptors that binds to the pathogen associated molecular patterns and play a role in recognizing a pathogen. So three class of pattern recognition receptors they are present. The first is the toll-like receptors. So toll-like receptors, they are the transmembrane receptors that are found on the immune cells like macrophages and dendritic cells. Scavengers receptors. The example of scavenger receptors are CD36, CD68 and SRB1. So they can bind to the component of the bacterial cell wall like lipopolysaccharides and peptidoglycan. The third are the mannose receptors. So these receptors, they are present on the surface of phagocytes. So the innate immune system, it recognizes the pathogen by identifying the specific molecular patterns that are known as PAMP, scavenger receptors and mannose receptors that play a key role in this process. So this recognition allows the innate immune system to distinguish between the pathogen and host own cells and mount a defense against the invaders. Now we can classify the antigen based on their ability to induce antibody formation into T cell dependent and T cell independent. T cell antigen require the involvement, the dependent antigens require the involvement of the T cells, specifically the helper T cells, to stimulate the production of antibodies by B cells. So these are typically more complex in structure, such as erythrocytes, serum proteins. They trigger the production of various types of antibodies including IgM, IgG, IgA and IgE. Whereas the T cell independent antigens, they can stimulate the production of antibodies by B cells without the involvement of the T cells. They are typically simpler, consisting of limited number of epitopes. Example are the lipopolysaccharides and flagellar protein, that is flagellae.